Today I will present MARS, the first in-storage processing system for raw signal genome analysis. We'll begin today's talk with a short background on genome analysis, then discuss our motivational goal. I walk you through the MARS architecture and our key techniques. And at the end, we'll look at the evaluation of performance and energy analysis. So genome analysis allows us to understand the genetic variation, evolution, and diseases, and thereby also enable breakthroughs in areas like outbreak tracing and personalized medicine. The process of genome analysis typically consists of two steps. The first one is sequencing, which produces raw electrical signals from DNA fragments. And the second one is mapping, where these fragments are then aligned to a reference genome to reconstruct the full genomic sequence. Traditionally, a deep learning step that's called base calling is used to translate these raw signals into the DNA sequence. However, this step is computationally very expensive, and we therefore focus on a more recent approach that's called raw signal genome analysis, or also RSGA, and bypasses this base calling step. Therefore, this also enables real-time analysis and leverages the high throughputs of modern nanopore sequences. The RSGA pipeline has two main phases. The first one is indexing, which happens offline, and mapping the raw signals then in an online step, which is also in focus of our work here. In the indexing phase, the reference genome is converted into event representations and then stored in a hash table for fast lookup. During the mapping phase, the raw electrical input signals from the sequencing are then mapped to the reference genome to reconstruct the full genomic sequence. And mapping can basically be split into three core steps. The first one is event detection, where the raw electrical signals are transformed into discrete events that represent segments of the signal. The second one is the seeding step, where events are compressed into hash values and then queried from the pre-built index hash table to find potential matches between the reference genome from the indexing step and the input sequence. And the last step is then called chaining, where the matching seed positions are grouped to form consistent and collinear regions that represent the final alignment. Let's now have a look at the motivation and the goal of our work. So as already indicated, sequencing throughput is increasing rapidly and also expected to increase in the future. However, RSGA pipelines struggle to keep up with these requirements, especially for larger genomes like the human genome, which is often in focus of the analysis. And we therefore identify the need for acceleration and started by identifying key bottlenecks in this RSGA pipeline. On the bottom, you can see the breakdown into the three key steps of mapping I just presented, as well as the I/O data movement overhead for 5 different real-world data sets, with D1 being the smallest and D5 the largest genome. And we observed that chaining and seeding dominate the runtime across all data sets, while also event detection and I/O overhead are significant for smaller genomes. And this motivated the need for us for efficient hardware acceleration, also deeper analysis of the accelerated pipeline. So we did a modeling experiment to understand the role of the I/O a bit more deeply, where we model a scenario where the seeding and chaining steps, so the last 2 steps of mapping, are accelerated. So here in this graph on the x-axis, you can see the degree of acceleration of these 2 steps, while on the y-axis, it displays the percentage of the I/O data movement as part of the end-to-end execution time. And you can see I/O quickly becomes the dominant bottleneck, accounting for up to 78%, even for the larger genomes, which is why we conclude that compute-only acceleration isn't sufficient, but we need to fundamentally reduce the data movement. Therefore, the goal of our work is to design a high-performant, energy-efficient, and scalable system for raw signal genome analysis by addressing the I/O data movement bottleneck as well as the computational bottlenecks we observed. Let's now have a closer look into the system we designed. So we introduced MARS, the first in-storage processing system for RSGA. And our key idea is to move the computation closer to where the data actually resides, so inside the SSD. And we also enable multiple processing in-memory paradigms within the SSD itself to create a highly parallel and energy-efficient system for raw signal genome analysis, thereby also exploiting the high SSD internal bandwidth and avoid host data transfers. MARS co-designs dedicated hardware inside the SSD with software modification that are targeted to optimize RSGA for hardware acceleration and storage. And as mentioned, we leverage processing using and near SSD internal DRAM paradigms for our design. We start by optimizing the RSGA software pipeline in two key ways to reduce the intermediate storage requirements, but also the computational load. The first thing we do is discard candidate matches between the raw signal input and the reference genome very early in the pipeline by using different filtering techniques, frequency and mode-based filtering. And the second thing we do is we do an arithmetic conversion from floating point to fixed point representations to reduce memory footprint and also computational costs. The main challenge in all of these changes is that we preserve the accuracy while doing that. And we therefore ensure careful placement of these techniques in the pipeline and fine-tune all the parameters that are involved. We also validate that these software improvements match the accuracy of state-of-the-art algorithms. And we evaluate this with the same 5 real-world genomic data sets of different sizes and compare our pipeline with either the filtering techniques only or the filtering techniques and the arithmetic conversions to the state-of-the-art pipeline that's called RowHash2. And we make two key observations. The first one is that our software pipelines actually outperforms recall end-of-1 scores over the RowHash2 algorithm across all data sets and also matches the precision values. So we therefore show that our filtering techniques are quite effective in eliminating ambiguous and also more redundant candidate matches and allow the pipeline to focus on more informative regions that are then also more likely to represent the correct match. The second observation we have is that also our fixed point arithmetic conversion only lead to a minimal accuracy loss. That shows that we have a careful usage of the normalization quantization within the pipeline, which are quite effective in keeping the accuracy. Let's now have a closer look into the actual hardware system we designed. Our system Mars operates in 2 modes. The first mode is the conventional mode, where the SSD continues to behave like a normal storage device. And the second mode is the accelerator mode, where then Mars takes over and executes the entire RSGA pipeline inside the SSD without any further host intervention. Let's now have a closer look at the components that make the system possible. The first one is the Mars control unit, which implements a final state machine that orchestrates the complete RSGA pipeline steps, including control and data flow. We then have dedicated sorter and merger units within the SSD controller. We have 1 per flash controller, so 8 in total in our design. And they can perform highly parallel hardware accelerated sorting and merging, which is a core step of the chaining step within read mapping. Additionally, we also modify the SSD internal DRAM. There we have 2 units. The first one is the arithmetic unit. There we have 1 per 2 subarrays, so 256 in total in our design. And they perform arithmetic and logical operations that are required for RSGA. And our design uses processing near memory. It's based on the Fulcrum design that leverages the DRAM subarray proximity for low latency parallel arithmetic operations. The second element we add is a querying unit per subarray, so 512 elements, our design, and they perform efficient in-memory hash table lookup for seeding where we need it. And this design that we implement here is based on Pluto that leverages the processing using memory paradigm to enable massively parallel hash table lookups directly within the DRAM. We then go ahead and break down the complete pipeline into smaller steps and then map them to each of the components. So we break them down, for example, arithmetic, querying, sorting operations, and then go ahead, map each of these tasks to the dedicated compute unit that can execute this paradigm and implement Mars as a pipeline. So for example, if there's a multiplication step, it would then be mapped to the arithmetic units in the DRAM. And last but not least, we ensure that the Mars control units encodes this pipeline to a final state machine and define the fixed execution sequence and then dynamically trigger the task as soon as the input is ready. Let's also have a quick look at the control and data flow of the application. So we start by loading the raw input signals from the flash memory to the DRAM to do the event detection step inside the arithmetic units. We then continue by triggering the hash value generation and filtering operations that are part of the seeding step, still in the arithmetic units, and continue with the hash table querying, that's still part of the seeding step, inside the querying units in the SSD internal DRAM. Next, we start with chaining, which start with a larger sorting step, where we create batches of sequences inside the arithmetic unit. We then transfer these unsorted sequences to the sorter units near the flash controller, where the shorter sequences are first sorted, and then these short sorted sequences are merged into longer sorted sequences by the merger unit. And this process continues until the sequence is fully sorted. And then transferred back to the control unit, where the last step of chaining is happening, which is a dynamic programming step. And therefore, there we also use the arithmetic units inside the SSD controller. Of course, we have many more details on the mechanism and also the system integration in the paper, so feel free to have a look at this. Let's now have a closer look into our evaluation results. So we compare our Mars design against 4 other state-of-the-art systems for performance and energy analysis. We use 2 baselines that are based on the base call-based pipeline, and then 2 that are based on the RSGA pipeline. The base call-based pipeline is first a conventional pipeline that uses GPU-based base calling with a Dorado-based caller, and then CPU-based read mapping with Minimap2. And then we also use a hardware-accelerated pipeline that uses the GenPIP design. That's a design that's based on non-volatile memory hardware acceleration, also for base calling and read mapping. We then also compare our design against 2 RSGA-based genome analysis pipelines. The first one is RowHash2, that's the state-of-the-art CPU-based RSGA pipeline. And then our own software pipeline also executed on the CPU as a comparison point to see how much influence the hardware acceleration has. For our hardware setup, we use a state-of-the-art server with 2 CPUs and 64 physical cores each, 1 terabyte of DDR4 DRAM, and for the base call baseline, also NVIDIA RTX A6000 GPU. The SSD that we use for our Mars system has 8 channels with 8 flash chips each. We use a PCIe 4.0 interface and 4 GB of SSD internal LPDDR4 DRAM with 16 banks and 512 subarrays. We also leverage widely used simulators for simulating all involved components in our designs and baselines. We start by conducting a performance evaluation, and we continue here to use the same 5 real-world genomic data sets that we used in the accuracy experiments. And here you can see the speed up of all baselines over the CPU-based RowHash2 implementation, so the RSGA state-of-the-art CPU implementation. And we observed that Mars outperforms all other baselines across all 5 data sets. And we make also 2 further observations. The first one is that we show that our software modifications provide a speed up of 1.7x over the RowHash2 baseline. And we therefore show that the modifications are effective in reducing the computational load. And additionally, we also show by outperforming GenPIP, which adopts processing in memory outside the storage, that we are fundamentally able to solve the I/O data movement overheads that was still visible there. Additionally, we also conduct an energy analysis where we show that Mars reduces energy consumption over all of the baselines across all data sets. Of course, part of it stems from the reduced execution time, but this mainly also comes from our storage-centric design that fundamentally addresses the I/O data movement overhead. So to conclude, we present Mars, the first in-storage system for raw signal genome analysis that's enabled by software improvements and the hardware design inside the storage subsystem. Our key design is to enable PIM paradigms within the SSD and leverage the high SSD internal bandwidth to create a highly parallel and energy efficient system for RSGA. We improve the performance over state-of-the-art software-based and hardware-based read mapping baselines, both in the base called and the RSGA domain, and also show that we significantly reduce energy consumptions over all of these baselines. We also show that our design matches the accuracy of base calling based pipelines and improves F1 score over the state-of-the-art RSGA pipeline. And our co-design of software and hardware entirely within the storage system therefore enables real-time, scalable, and energy-efficient RSGA. With that, I would like to thank you for your attention, and I'm happy to take any questions.
Hi, very nice work. So I'm curious, just in general, a very general question about your application. So in bioinformatics, basically, there's always partial matching, and that's where an immense amount of hardware usually goes. Is this part of what you're doing before that step?
So this work focused completely on the read mapping pipeline, and part of the chaining step is also matching the input sequences to the reference genome. So there's kind of similar paradigms that are included in this step. There are other works that also then continue with alignment, but we already are able to achieve very high accuracy results, which is why we didn't include it here for now.
So it's like pre-processing step.
Kind of, yeah.
The other question is that, of course, people in FPGA people have been sticking FPGAs onto I/O devices to do this sort of work for 20 years or more. I'm curious whether your group has any interest in that or has done comparisons.
Like, you mean compared to other storage, near storage work?
Just goes way back into obvious things that FPGAs are very good at handling complex streaming processes. And so there have been products where FPGAs have been put into disk drives to do this sort of work.
Yeah.
And that was a long time ago, and advanced since then.
Yeah, I mean, we definitely looked into all of these different works that are out there, and also our work. Our group is focusing on other work related to in storage acceleration, also FPGA-based acceleration, and we actually also compared this work against a setup with a smart SSD, where we have the direct connect between the storage and the FPGA, and we moved the sorting part to the smart SSD, and we're also able to outperform it as well. Yeah, so unfortunately not enough time, but we also showed this ablation study in our paper. Yeah.
So I was actually quite curious about the sorting part. The way you're showing in your picture, it almost seems like you're sorting on the way from the memory into flash. But doesn't only give you a partial sort unless you do multiple round trips to the memory?
Yeah, so so it depends on the length of the sorting, that's true. So in this step that is in chaining, there are actually a few sorts that are, like, I would say, of medium size, so a few kilobytes. And for those, we have registers near the mergers such that we can do round trips there. And if it should be a longer sequence, then we actually transfer it back to the DRAM, and we hide the latency, the data movement latency, by basically placing it such that we always have the data ready if we want to merge.
Yeah, thank you.
More questions?
Yeah, one. You showed a picture that was using the DRAM of your flash and the flash controller. You, at some phase, you were exchanging data from between the controller and the DRAM. Is that a bottleneck?
So it isn't a bottleneck because we hide the latency by the computation. So that's actually quite a good thing that we can do this within the sorter. And we use the Mars control unit to ensure that large part of the latency is hidden. Yeah.
Thank you very much. Let's take a again.